Hello and welcome back to the BASATS channel, the Biochemistry for Health Sciences channel. In the previous videos, we talked about carbohydrates and the importance as fuel sources, especially glucose. And then we talked about carbohydrates as identification molecules for molecular recognition. Today we'll give one more example of carbohydrates as identification molecules, and that's the ABO blood groups that we find in humans on red blood cells. So in the previous video we talked about why carbohydrates are very, very good molecules for molecular recognition. Basically, molecular recognition, which is basically identification, ID molecules. One is there are so many different types of monosaccharides, and the second reason why these are very good for making ID molecules is because two monosaccharides can be attached to one another in many, many different ways. Okay, so we give the example of the hexasaccharide. If you join 6 carbohydrates together, you could have billions of ways of doing that. Joining 6 nucleotides together in a nucleic acid, you only have about 4,000 ways of doing that. Joining 6 amino acids together in a protein, you have only 86 million ways of doing that. Okay, so clearly billions of ways means billions of shapes. And in shapes, in biology, shapes is also identification. So one of the examples of carbohydrates for identification, these are surface markers that are present on red blood cells, Okay, so you have this carbohydrates, these oligosaccharides that are present on red blood cells that give you the ABO groups. So all of these groups, type O, type A, type B, they all have the basic oligosaccharide. They all have the common basic oligosaccharide. But in type A, there is an additional monosaccharide, N-acetyl galactosamine, while in type B, there is an additional monosaccharide, galactose. Okay, so this is type O, which is the basic monosaccharide sequence, and then the type A and the type B. So people with red blood cells of type O, this is what you see on their surface. Type O red blood cells, type A red blood cells, this is what you see on the surface. And people with type B, you will see this. Now, people with type AB, you will see both the A as well as the B antigen present on their red blood cells. So just to summarize, person with type A, their red blood cells will have antigen A. So this is what you see on their red blood cells. Their plasma will contain the antibody against B, against antigen B, not A. Because if they had the antibody against A, then their antibody will react with their own antigen and destroy their red blood cells. So that's why they have anti-B. Person with type B will have B antigen on their red blood cells. They will have antibody against A. Okay, so you can see it's logical. People with type AB will have on their surface type A as well as type B antigen, the A antigen and the B antigen. So their plasma should not have any antibodies that react with antigen A or antigen B. On the other hand, people with type O, let me erase this. Okay, so people with type O will not have any of these A or B antigens on the surface. So they won't have any of this, but that means they will have antibodies against A and antibodies against B. So that will be type O. So you can see from this sequence here, from this table, that a type A person can donate RBCs. And RBC donation is a very common thing we do in blood transfusion. As you know, red blood cells carry oxygen, and oxygen is critical for energy survival, for energy production for survival. So this is critical. So type A can donate red blood cells to people with type A blood, because those who have type A blood have anti-B, and they can donate to type AB because they do not have anti-A. Okay, neither do these have anti-A, neither do these have the antibodies against A, so that's why they can donate to their same type A or to AB. Same logic, B can only donate to B or AB because ABs have no antibodies. So they can take, ABs can get blood from either from A or B. Those with type AB, they can donate their red blood cells to only AB. Okay, because AB do not have antibody against A or against B. However, their plasma, because they don't have antibodies, they can donate their plasma to anyone since they don't have antibodies. So type AB is the universal plasma donor. Type O, they can donate their red blood cells to everyone because they do not have the A antigen and they do not have the B antigen. So type O are the universal RBC donors. So the ABO then is another example here, a very nice clinical example of carbohydrates being used as a identity tag. Various red blood cells. So basically all red blood cells are not the same. We cannot simply transfuse one person's red blood cells into another person's body. Okay, so we have to do this blood typing, and that is done in the immunohematology lab. We'll talk about the other antigens such as the D antigen or the Rh0 in some other videos. But this is very important. The blood has to be typed to make sure that the blood is compatible from the donor to the host. Okay, so this is very important. So you can see there, if you consider the ABO as well as the D antigen, you can have all kinds of blood typing. A plus means they have a Rh antigen. A minus, A without the Rh antigen. Similarly, B plus, B minus, O plus, O minus, AB plus, AB minus. The O minus is best for emergency transfusions of red blood cells because they can donate to anyone. O plus is the most common, and AB minus is the least common that we see in the population. So once again, carbohydrates, keep that in mind. Yes, they are very important for fuel, which is required for energy. And the second important function of carbohydrates is identification, which is important for molecular recognition. Okay, now with why these different red blood cells have these different types, type A, type B, type O, we are not certain, but there are recent articles that do talk about the relationship between the ABO types as well as the association with the type of microbes that you find in the colon. Okay, so this is more recent articles whether certain microbes prefer to see certain antigens, and that's why they colonize the colon. This is very initial research. Hopefully we will get more information about this. And remember, this area, the importance of microbes in our colon is getting more and more attention. And this will be a very important area that healthcare professionals will have to consider when we treat human diseases. So let's stop here, and hopefully we will see you in the next video. Goodbye.